So the topic of today's forum is design and development of engineered scaffold proteins for targeted diagnostics and therapy of cancer. And this project is a collaborative project between University of Uppsala University in Sweden, Chemical University in Moscow, Tomsk Polytechnic University, and the Tomsk National Research Medical Center. And this project is supported by a grant of Ministry of Science and Higher Education of Russian Federation. And I think this is a short program. We will have a welcoming word from Vladimir Tolmachev, who is a professor at Uppsala University and also head of the Diagnostics Center in Tomsk, followed by welcoming words from Sergei Mikhailovich, who is a professor and a member of Russian Academy of Science from Moscow, and then followed by presentations of three researchers. Olga Bragina, who is a medical doctor and a PhD, will talk about phase 1 clinical trial using a technetium labeled ADAPT6 scaffold protein from Tomsk, followed by me talking about DARPins for radionuclide molecular imaging, and followed by Olga Shilova, who is a PhD and a researcher, will talk about HER2 targeted anti-cancer therapeutics from Moscow. Please, Vladimir, you're welcome to give a few welcoming words.
Thank you, Angelica. Dear colleagues, dear friends, I'm extremely happy to greet you at this first Young Researcher Forum. This forum has short but very saturated prehistory. A little bit more than 3 years, in late June of 2017, we meet at Radio Pharma Forum with Professor Chernov, who asked to help a little bit with preclinical characterization of labeled DARPins. The same week we met with Professor Dev, and I think we had extremely good chemistry with those researchers, and it resulted in impressive development. Now, our constellation includes 5 research centers in 2 countries, and this work was boosted a lot by grant of government of Russian Federation, which permit us to proceed to clinical study and establish Oncotheranostic Center based on the Tomsk Polytechnic University. The formal results for the moment of this collaboration are 10 papers, joint papers in good international journals. But more important, we have a team of researchers who are capable to solve any degree of complexity, problems with any degree of complexity spanning from biotechnology, preclinical development and characterization, and performing clinical studies. The most important is that we have a constellation of bright, talented and hardworking young researchers. This is particularly important because this means that this work will continue and hopefully over decades. Today we're meeting together first time face to face to learn the faces on behind the names of our joint papers. I hope that it will be a story of success, and I hope that this will continue, and it's just a first meeting in a long series of exciting scientific events. You are very welcome. Thank you very much.
Thank you very much, Vladimir, for this great welcoming words. I think we can give a word to Sergei Mikhailovich Deev.
Dear colleagues, dear friends, all important points were already mentioned by Professor Tolmachев. I just would like to add a couple of words. My vision of this project is extremely good. To be honest, I'm involved in the expertise of many projects, also mega grants, and I can say that this one is extremely interesting because it's not only great scientific results, but also we have a real perspective for translation results in the clinical practice. I am really be happy to have this fantastic contact with Professor Tolmachev team, as well with Professor Chernov team, because it's really a very good alloy of the great fundamental science, chemistry, biology, and real medicine. So young scientists could be very enthusiastic and could be involved in this translation to the real medicine. Thank you so for your attention for my speech. Thank you for your participation in this conference, and a lot of thanks for your enthusiasm to involve your efforts in the real results of our joint project. Thank you.
Thank you very much, Sergei Mikhailovich, and I think we can move to the first speaker, who will be Olga Bragina. She will talk about the phase 1 clinical trial using radio-labeled ADAP6 scaffold protein, and I will stop sharing my screen, and you can start to share your screen. Just to unmute your microphone because we cannot hear you. Yes.
Do you hear me right now? Yes, probably. That's right.
Подожди, она как демонстрируется, так или?
Нет. Yeah, hello again, dear colleague, and I really like to see you all here, and I very appreciate to Vladimir Maximilianovich and Sergei Mikhailovich for opportunity to put forward our results here on first Oncotheranostic Forum. In this report, I will be talking about first clinical experience of using target ADAP6 molecules labeled with technetium-99M for diagnosis of HER2-positive breast cancer. So for many years, a personalized approach has been widely used in the treatment of oncological disease and characterized by a therapy according to tumor molecular profile to obtain the better response. Despite the large number of molecular targets, much attention is still being paid to the study of the surface human epidermal growth factor type 2 or HER2 neo receptor. I would like to pay attention not on molecular or biological characteristic of this receptor, but mainly focus on his clinical features. First of all, I want to mention a wide variability of oncological disease with overexpression of HER2, and of course, mainly in breast cancer patient, ovarian cancer, and gastric cancer. The main point is when we talk about the HER2 positive breast cancer patient, we should talk about aggressive form of the disease and high rate of metastasis in this localization, and also shortened disease-free and overall survival, and also recurrence rate. The most important that the patient with the HER2 positive breast cancer, they need a targeted therapy which extremely improve prognosis of the disease. And nowadays, where 4 medicine at least are used in oncological practice, widely used in oncological practice for treatment of breast cancer with overexpression of HER2-neo in monotherapy or in combination with chemotherapy. There are a number of diagnostic techniques for determination of HER2 status and HER2 expression. Also used methods of in situ hybridization such as fluorescence hybridization or homogenic hybridization in situ for which detect amplification of HER2 gene. Unfortunately, they have their disadvantages. And for example, invasive manipulation, I mean core biopsy or operation. The second one is the subjective factor, I mean the violation of research methodology and result interpretation. Also, with positive and false negative test results, mostly in immunohistochemistry. Impossibility of tumor spread in vivo assessment. I talk about the primary tumor, lymph nodes, and distant organs and tissues, and intratumoral heterogeneity. The most important, I think, it's when we talk about the discordance in HER2 status in primary tumor, lymph node metastasis, and distant metastasis. And it's about figures from 10 to 25%. In recent years, radioisotope methods for specific tumor detection have been actively developed using various of antibodies and radionuclides. Currently, instead of full-length antibodies, molecular structure or alternative scaffold protein are used and have all requirements for optimal radionuclide delivery to tumor cells. The first target proteins molecules used in clinical practice was the Ophi-body, and here a part of the clinical study of molecules labeled with gallium-69 68 for PET diagnosis of HER2 positive breast cancer, which were performed in Sweden. In particular, this is the case with the HER2 negative primary tumor and HER2 positive metastatic site in liver. Nowadays, in collaboration with Tomsk Cancer Research Institute, Institute of Bioorganic Chemistry, Tomsk Polytechnic, and Uppsala University, we have performed clinical studies of some scaffold proteins. And one of them is ADAP6 protein, which has high affinity to HER2, of target interaction, rapid clearance from blood, high specificity, and low immunogenicity. As a radionuclide, we use technetium-99M, which has a short half-life, the absence of beta radiation, and low cost and simplicity of technetium-99M obtaining. There are 29 breast cancer patients were included in this study. Before the treatment, all clinical and instrumental studies were performed, also as morphological and HER2 neo detection for biopsy samples of primary tumors. The patients were injected with 500 or 1,250 microgram of ADAP6. The characterization of each group are presented on the slide. Labeling of ADAP6 was performed in aseptic condition in nuclear medicine department of Tomsk Cancer Research Institute just before use and injected intravenously. Anterior and posterior planar whole body imaging and SPECT scans were performed at 2, 4, 6, and 24 hours after injection. In results, no drug-related adverse reaction or changes in vital sign were observed during imaging or follow-up period. Also, as no changes in blood or urine analysis were detected. The highest uptake in normal organs were observed in kidneys, liver, and lungs. Moderate activity was observed in the gastrointestinal tract. Distribution of activity was similar after injection of 500 and 1000 microgram. The only significant difference was in uptake in intestinal content at 6 and 24 hours after injection, which was lower for 500 microgram. When we talk about the blood kinetics, the elimination rate for 500 and 1000 microgram were similar. The highest absorbed organ dose was in the kidney. Doses to adrenal, gallbladder, wall, liver, spleen, and pancreas were also noticeable, but they were several-fold lower than the renal dose. Doses to adrenal, stomach, wall, spleen, thyroid, and uterus were significantly higher for 1000 microgram dose, but absolute difference was prominent only for adrenal and thyroid. Total effective dose was 0.009 for 500 microgram subgroup and 0.01 for 1000 microgram group. The best discrimination between tumors with high and low HER2 expression was provided by injection of 500 microgram protein. There, the mean value of tumor to contralateral breast ratio for HER2 positive breast cancer tumor was 37, already 2 hours after injection, significantly higher than the value for HER2 negative tumors. The ratio tended to increase with time. The tumor to contralateral breast ratio for 500 microgram group was significantly higher than for 1000 microgram group. Moreover, the difference in tumor to contralateral breast ratio value between HER positive and HER2 negative tumors in case of injection of 1000 microgram was not significant at any time point. Also, the tumor to contralateral breast ratio for 250 microgram at 2 hours after injection was also significantly lower than for 500 microgram. Injection of 500 or 1000 microgram technetium-99 ADAPT6 enabled higher uptake in tumors than in liver. There was a tendency of higher tumor to liver ratios for the 500 microgram proteins group. But the difference between ratio for doses of 500 and 1000 microgram was not significant. Injection of 250 microgram resulted in significantly lower tumor to liver uptake than injection of 500 microgram group. Here also I want to focus on two interesting clinical cases which were identified in this study. The first is the HER2 positive breast cancer patient by immunohistochemistry, but during our study we revealed low tumor to contralateral breast ratio at 2 hours. The biopsy sample were reevaluated by pathologist and the HER2 status were reassigned as 2+ and FISH negative. Based on the new pathology evaluation here, treatment were changed by the continuation of HER2 targeting therapy, and as I remember, it was combination of trastuzumab and pertuzumab target medicine. The second case I want to mention is the case of HER2 positive breast cancer in patient with the primary tumor, the big primary tumor, and involved metastatic lymph nodes. And during our study, we revealed the metastatic site were revealed in 5 rib and thoracic vertebra 9 and 8. During their examination, CT imaging and bone scan using technetium-99 pyrophosphate didn't reveal any metastasis. So it's a picture of bone scan, and here the MRI imaging which were performed after our clinical examination, and MRI imaging revealed and confirmed metastasis in thoracic 9 and 8 vertebra. The treatment strategy of this patient was changed also from surgical treatment to chemotherapy in combination with targeted therapy. And after 6 months after study with the use of technetium-99m DAP6 on their bone scan with technetium-99 pyrophosphate and CT imaging were revealed their metastatic sites on the 5 rib and thoracic vertebra 9 and 8. In conclusion, I would like to say that technetium-99m DAP6 are safe and well tolerated. Injection of 500 microgram provide excellent discrimination already after injection, and the tumor to contralateral ratio had a tendency to increase this time. 500 microgram dose appears optimal, and deviation from this dose dose would result in decrease of both sensitivity and specificity of imaging of HER2 expression. Thank you all, and sorry in advance for my answering question, which absolutely won't be good enough. Yeah, thank you again.
Thank you very much, Olga, for your great presentation and a great talk. I would like also to say that if you have questions, you can type them in the chat, and afterwards we can read out if you would have any questions. But of course, right now it's open to all participants to ask questions.
May I ask a question?
Yes, of course.
Olga, first of all, great talk. Thank you very much. I'm pleased to see this presentation. What is your vision how to proceed with those studies?
I'm sorry, how to proceed?
How to proceed? Are you happy with available data, or you want to test more tracers? Or
yes, I'm very happy. Actually, I'm very proud to be a part of this wonderful team, and in clinical studies, I mean, and to be a part of experimental study, and I think it's a great collaboration. And I also happy to study a lot of tracers, so I think it's more than I have in my presentation. It's it's about 4, but in practice, I think it's more than 6 tracers, and of course, it's a great opportunity to study them. Thank you.
thank you.
Would any of the participants like to ask any questions? Maybe I can ask a question. Do you think the number of patients was enough, or would you like to include more patients in the study?
I think that the number of patients were enough for the first phase of the clinical study. So I think that it's not the end, and a lot of thoughts about the continue of ADEPT-6 clinical examination. So I think for the first study, it were more than enough.
Okay, great. Then probably we can continue with the presentation, and if anyone will have questions, you're welcome to type them in the chat, and we will read them, of course. So then I guess I would present the work that we did in here in Uppsala. I would just like to ask to unshare your screen so that I can share the presentation.
Olga,
Oh.
stop sharing screen.
Awesome.
Green.
I'm sorry.
Запустили. Остановить. Окей.
So I would like to present a summary of all the work that we did more or less over the last 3 years. It's the project. There are several projects that we did, and they all based about the development of DARPin-based probes for radionuclide molecular imaging. And this was a collaboration between Institute of Organic Chemistry in Moscow, where the protein production was performed. Afterwards, the proteins were analyzed in the KTH in Stockholm. In Uppsala, we did the radiolabeling work in vitro and in vivo validation. And the results of this preclinical validation were as a basis for clinical translation in Tomsk. So first, I would just like to talk about DARPins. They are a new class of targeting probes, and they are much smaller than the antibodies. So probably you have all heard about antibodies. They have been traditionally used for imaging, but they are very large. So they are 150 kilodalton in size, and due to this large size, they have a long half-life in blood. So they circulate in the blood for several weeks. And of course, for imaging, it's not so good because you will have very high activity in blood and low imaging contrast. So on this picture, you can see the imaging obtained after injection of 89 zirconium labeled trastuzumab. And even 5 days after injection, you can see the activity in the main blood vessels in the body. And of course, the imaging contrast is quite low. Moreover, antibodies are quite sensitive biological molecules. They are sensitive to temperature, so they cannot be heated more than 37 or 40 degrees. And they're also very sensitive to pH changes, which means that we are very limited to the number of methods that can be used to radiolabel biological molecules. And in contrast to monoclonal antibodies, DARPins are about 10 times smaller in size. So depending on the structure, they can be 14 to 18 kilodaltons. And due to their small size, they have a very rapid pharmacokinetics, so they are excreted from the blood shortly after injection, which means that you can obtain quite high imaging contrast just several hours after injection. And DARPins consist of a quite big library of various binders, and they have high affinity to targets. And they're also rather robust, at least in comparison with the antibodies. They can tolerate heating at elevated temperatures and also different changes in pH, which means that we can use various labeling methods. And the target that we started to work with was HER2. So HER2 is overexpressed in breast, the gastroesophageal or ovarian cancers, and it's also a target for a targeted therapy with trastuzumab or a conjugate of trastuzumab with the cytotoxic drug DM1. And imaging of HER2 allows to evaluate expression of this target in the whole body, non-invasively, allows to select patients for therapy and monitor their response to treatment. And we have used, we have studied two DARPins that bind to HER2. One is called 929 and another one is DARPin G3. DARPin 929 consists of 5 antiring repeats, and each repeat consists of about 33 amino acids. So the size of 929 is higher than of DARPin G3. However, the affinity of 929 to HER2 is lower than the affinity of G3. And a little bit about the choice of the radionuclide. We have considered technetium-99M and various isotopes of iodine. In the lab, we work with iodine-125 due to convenience and the long half-life. However, there are also isotopes of iodine-123 that can be used for SPECT and iodine-124 that can be used for PET. And the reason why we chose this nuclide, first of all, we considered availability of both nuclides and also of imaging modality. The SPECT is an imaging modality that is still the most commonly used worldwide. About 80% of diagnostic scans performed in the world using SPECT. And technetium is readily available from generator, and production of iodine-123 is possible in Tomsk cyclotron. The second factor that we considered was to have a relatively straightforward labeling. We wanted to have the radiopharmaceutical for clinical use that can be prepared as fast and as simple as possible. And we have considered tricarbonyl technetium because it can be prepared using commercially available kits, and the labeling can be, attachment of tricarbonyl can be performed to histidine, and histidine can be encoded in the protein and form so-called peptide chelator. And the direct iodination is also robust and straightforward procedure. And the third factor is quite interesting. We decided to compare tricarbonyl technetium label because it has residualizing properties, and iodine label has non-residualizing properties. And on next slide, I would like to explain a little bit more in detail what it means. So the actually residualizing and non-residualizing labels are processed differently by living systems. And on this short graph, you can see the radiolabeled protein when it bound to the receptor on the surface of cells. It can be a cancer cell, but it can also be a cell in the normal organs like kidney cells or liver cells. So after binding to the receptor, the radiolabeled protein goes inside the cell in the endosome, and then it forms, it goes into the lysosome, and under the enzyme action, the radiolabeled protein is chopped in different fragments that still carry the radioactive label. Depending on the nature of this radio catabolites, the catabolites either stay inside the cell or they are excreted outside of the cell. And iodine catabolites have non-residualizing properties because they are non-charging lipophilic, so they can go through the membranes because the membranes are also lipophilic, so they would be excreted from the cells quite fast, or technetium tricarbonyl metabolites have residualizing properties, so they are charged and quite hydrophilic, so they stay inside the cell much longer. And there is also a difference between internalization process in tumors and in normal organs. And of course, the imaging contrast depends on the signal in the tumor compared to the signal of the background or to the normal in the normal tissues. And it is quite important to understand the speed of internalization in tumor, because that would mean what kind of label can be used. So first we started with first anti-HER2 DARPin, which was DARPin 929, and we have labeled it with the tricarbonyl technetium to the hexahistidine tag, and we use direct iodination of tyrosine residues in the DARPin. And we have seen that both labels provide quite good visualization of the tumor. We have also seen that direct iodination, due to these non-visualizing properties of the catabolite, provides quite good imaging contrast. So the activity from normal organs is rapidly excreted from the body. And with the tricarbonyl technetium, which is a residualizing label, we have seen that there is a quite large uptake in liver and gastrointestinal tract. It also can be due to the lipophilic character of the label itself. So the combination of tricarbonyl technetium with hexahistidine provides a rather lipophilic complex. So we have later wanted to compare the DARPin 929 with the DARPin G3, because we knew that the DARPin G3 has a higher affinity to HER2. So we have performed a comparison, side-by-side comparison of two DARPins 929 with G3 labeled with technetium and iodine. We have indeed saw that the DARPin G3 has a higher affinity, about 100 picomolar to HER2. We have also compared their internalization by cancer cells in vitro, and we saw that the DARPin 929 has quite high internalization, about 50% after 24 hours, but DARPin G3 has a slightly lower internalization, about 25%. So what it means when we look at the in vivo situation, at the tumor uptake in vivo, we could see that the iodine label leaks out much faster from the tumor at 24 hours compared to the iodine label on DARPin G3. So the label stays longer and there is a higher retention. So due to high internalization of 929, iodine label leaks from the tumor all the time. And because DARPin G3 has a lower internalization, both technetium and iodine labels are suitable for this DARPin. And this is a comparative imaging of two DARPins. And we could see that the DARPin G3 provides about 4 times higher tumor uptake and also tumor retention, much better imaging contrast compared to 929, and the tricarbonyl label provided 4 times lower liver uptake than the DARPin 929, and also provided much higher tumor to organ ratios and better imaging contrast. So we have found that the DARPin G3 was better than 929 due to its smaller size and also higher affinity to HER2. And in our next study, we wanted to further improve the tricarbonyl label on the DARPin G3. And in this work, we have compared various histidine chelators. So from the previous work from antibody molecules, we knew that introduction of a glutamic acid residue in the chelator helps to lower the lipophilicity. So in this work, we have compared a position and a composition of a histidine-based chelator. So we have compared N-terminal and C-terminal position, and also hexahistidine versus histidine glutamate-based chelator. And what we found is that the N-terminal position of the HHH tag for tricarbonyl technetium label provided the lowest uptake in the liver, can be seen on these images, and also the tumor uptake was the same for all 4 variants, but the liver uptake was the lowest for the N-terminal variant, and it provided the highest contrast to liver and also highest the tumor to liver ratio. And in our next series of work, we decided to optimize the iodine label for the DARPin-3. So we started with the indirect labeling method using this HPM hydroxyphenylethyl label. We wanted to have a site-specific labeling method, so we used a G3 DARPin having a glycine, and a cysteine residue. However, this combination together with HPM was not very good. So what we saw in black here is the activity in the intestinal content, which indicates rather high lipophilicity of this combination, and it means that the activity would be taken up in liver, secreted with the bile to the intestines, and also the tumor uptake was much lower compared to the directly labeled G3. So then we moved on to another type of label called para-iodobenzoate label. It's also indirect method, and we have compared it with a direct method to the tyrosines, and we have seen that indirect labeling using TID provides much higher imaging contrast. At 3 hours post-injection, we see some activity in the kidneys, but by 6 hours, the activity from the kidneys is gone, and the main site of activity accumulation is in the tumor. And the direct labeling, the one feature of the direct labeling method is the accumulation of radiocatabolites in the thyroid and stomach. However, this can be blocked in clinics by the using of cold iodide or Lugol solution. And on this slide, I would just like to quickly show some results that were obtained from the clinical translation of 2 DARPins, 929 and G3. So both DARPins were investigated in the clinical trials, and both DARPins were able to visualize HER2 expressing metastases, and the dosimetry was better than for PET probes. And further, I would like to show a few research results that we obtained working with the second target. It's epithelial cell adhesion molecule, or EpCAM. What is very interesting about this target is its pancarcinoma antigen, which means it's overexpressed in several types of cancer, quite high expression in ovarian cancer, prostate, and also pancreatic cancer. And actually, there were 2 imaging agents, and one imaging agent for EpCAM was even approved by FDA in 1998, and it's called Verluma, or 99 listing labeled fat fragment. However, this agent had a quite high hepatobiliary excretion, as can be seen on this image. And the second agent was based on a monoclonal antibody called MOX31, labeled with indium for small cell lung cancer imaging. However, it also had quite high liver uptake and due to instability of the label. And the aim was to use this imaging probe for theranostic applications, just as for HER2, as I mentioned previously. So we have started working with the DARPin EC1 for imaging of EpCAM. It consists of 5 unkilling repeats, and it has an affinity of 18 kilodalton and quite high affinity, a molecular weight of 18 kilodalton and quite high affinity to EpCAM, 70 picomolar. And we wanted to investigate if the hypothesis that if internalization of DARPin EC1 by cancer cells is slow, then we could use both residualizing and non-residualizing labels. So as non-residualizing labels, we compared indirect PID label and the direct label. And as a residualizing label, we compared hexahistidine tag with the histidine glutamyl tag. We have compared the cellular processing of the 4 different variants by pancreatic cancer cells. What we saw was a rather slow internalization of DARPin EC1. Only 25% of DARPin was internalized after 24 hours, and that was the first indication that perhaps we could use both non-residualizing and residualizing labels. And we have performed an in vivo comparison in a mouse model of having the XPC3 xenograft. What we could see is the rapid renal clearance of DARPin. We also saw that the HEHE tag provides much lower uptake in the liver compared to the hexahistidine tag, which is the red. And all 4 variants actually had similar tumor uptake. However, the indirect label using PID, which is in blue, provided the lowest uptake in normal organs. The only organ with a high uptake were kidneys. And it also provided the highest tumor-to-organ ratios compared to all other labeling methods. And the tumor-to-organ ratios further increased by 6 hours post-injection. And if we compare our data with the literature data, considering that there was a different tumor model, but comparing our agents with the Verluma that was approved by FDA, we could see that already at 3 hours, DARPin provided high tumor-to-organ ratios, tumor-to-blood, intestine, and muscle, and even higher tumor-to-organ ratios by 6 hours. We have also shown that the tumor uptake is very specific by comparing the uptake in the outcome-negative tumor model ramas, and we also showed that the uptake is saturable and blockable by an excess of the cold DARPin. And we could demonstrate that the indirect label provided much higher imaging contrast compared to the direct label. We have then did a comparison we did a comparison, or we did a study in several cancer xenografts, in ovarian cancer xenografts, in OSCAR3 and SCOF3 tumor models, as well as in triple-negative breast cancer xenografts, and we could demonstrate that the DARPin was able to visualize outcome expression in various cancer xenografts. Right now, we have an ongoing study where we actually evaluate what is the optimal labeling position for the DARPin EC1. Is it labeled at N-terminus of the protein or at C-terminus of the protein? And we have also investigated what is the optimal composition of the label. Is it cobalt-57, gallium-68, indium-111, and a non-residualizing label using HPM. So what is quite interesting is here we have gallium-68, which is a PET nuclide, indium, which is a SPECT nuclide, cobalt-57 was as a surrogate for cobalt-55, which can also and HPM iodine can be iodine-123 or iodine-124. And in conclusion, we demonstrated that if a DARPin-based probe is slowly internalized by cancer cells, then a non-residualizing label is preferable because it provides much higher imaging contrast due to the fast excretion from normal organs and high retention in the tumor. We shown that indirect iodination using PID is the optimal strategy, and it provided high imaging contrast in several outcome expressing xenografts. And in clinical practice, labeling with iodine 123 for SPECT imaging can be envisioned. And concluding the series of studies, we have shown that having an optimized format of DARPin provides quite high improvement in the imaging contrast, and also having the right labeling chemistry is quite important, because it can also lead to very big improvement in the imaging contrast. So optimization of both scaffold composition and labeling chemistry is very essential. And this is the short summary of our published work during this collaboration. And I'm very happy, and I'm looking forward to further studies that are currently ongoing and also planned for the future for this very exciting project. And I would like to thank all our collaborators from Russia, from Moscow, and from Tomsk, also from Stockholm, and of course here in Uppsala we have a great team. And I would like to thank you for your attention.
Great. Do we have any questions?
I would have probably question to Doctors Chernov and Bragina. Have Angelica convinced you that working with iodine is of interest?
Yes, we are interested in working with iodine, and now we have all that we possible to get license for iodine 123, and also we work with our colleagues in Polytechnical University to get enough iodine 123, and I hope that in the beginning of next year we will have opportunity to work with the radiopharmaceutical. I already discussed with our colleagues in gynecology, oncogynecology department, and we have consensus to get the patient with the.
You mean ovarian cancer?
Yes, yes, in ovarian cancer.
HER2-positive ovarian cancer patient.
All right, so we'll go on.
So I think we will continue to the presentation of Olga Shilova, who will talk about HER2-targeted anti-cancer therapeutics from Moscow.
Thank you so much. Now I'll try to share my screen. I hope you can see it well. Can you see it?
It's loading, I think.
Loading. Okay. I hope it will work well in a minute or so.
Okay, now it works.
Oh, okay. Thank you so much for the opportunity to present our results. Maybe they are a little bit more modest, but I think they can complement well the whole study, because our work was devoted to different effector module, namely the protein therapeutics. I'll try to present you our work that was devoted to DARPins. As we have also already mentioned, till the last couple of decades, the targeted therapy of cancer was mainly based on antibodies. The variable fragment was and is used for targeted recognition, and sometimes the interference with signaling. And the constant part is used for activation of immune cells and molecules, and also is very actively used for toxic component delivery. But during the last 20 years, a variety of both targeting and effector modules have been developed. And the choice of toxic moieties for cancer therapy is very wide. It varies from traditional chemotherapy drugs and highly efficient radionuclides to photosensitizers and some protein toxins, like toxins that act on translation, for example, diphtheria toxin and exotoxin, Pseudomonas exotoxin A, and some highly efficient ribonucleases like RNase, vinase, and RNase A. The choice of effector would depend on complex circumstances, including, of course, tumor biology, the features of the target molecule, and also, what is very important, the compatibility with the targeting module. Today we have already heard about DARPins, these artificial targeting proteins that consist of anchoring repeats. And I would just mention that they have some very favorable features, namely their small size, their stability in wide range of pH and temperature values, and they are also very easily produced in bacteria. So they can be included in fused proteins, and these hybrid molecules can be produced as a single agent with known stoichiometry. In our work, we used DARPin 929 and started with the module protein that consisted of DARPin and fluorescent protein and cherry. We found that it is highly efficiently produced in bacteria, and in this protein, DARPin retains its specificity and high affinity to HER2 receptor, which is confirmed both by flow cytometry and fluorescent microscopy. So as a next step, we tried to combine DARPin with different protein toxins. We started with protein photosensitizer MiniSOC. It also was efficiently produced, and it did cause specific cell death in HER2 expressing cells. In this model, we used breast adenocarcinoma cells, SKBR3, and we knew that the effect of MiniSOC with blue light was intermediated by oxidative stress, because the adding of antioxidant, namely ascorbic acid, decreased the toxicity. It is the predominant mechanism of DARPin MiniSOC toxicity was necrosis. We can prove it with early cell membrane damage, which is indicated by crepidemiodine stain, the typical cell morphology, and also the absence of DNA fragmentation, what is typical for apoptosis, another cell death type that is probable for oxidative stress response. What is even more interesting is that MiniSOC has its own fluorescent features, so we can trace its destiny in the cell. We know that DARPin MiniSOC binds to HER2 on cell surface. Then, in physiological temperature, it internalizes and goes to endosome. And after 2 hours, its signal colocalizes with the signal of fluorescently labeled lysosomes. And it also should be noted that the intensity of fluorescence decreases when minisoc is being internalized. It means that we can trace the rate of internalization with the flow cytometry. What was made for two proteins, the mentioned DARPin minisoc and a very similar protein that contained another targeting module, the single chain antibody 45. As we can see, at low temperature, the whole internalization is inhibited and no fluorescence decrease happens. But if we incubate these proteins with cells at physiological temperature, the decrease of fluorescence occurs, and the rate of this process is higher in case of DARPin minisoc complex with HER2. It means that DARPin 929 is efficiently internalized, and I'm happy that this data correspond to the ones obtained by radio labeling. This means that the combining of DARPin 929 with minisoc is not very favorable because the internalization seems to decrease the efficiency of minisoc for the sensitizer. But we can use this feature to deliver some proteins that need to be internalized to cytosol for their process of action. As an example, we can use the Pseudomonas aeruginosa exotoxin A. Further, it would be denoted as PE. This is the protein that has several domains, and it helps to use the truncated variants of this protein that lack natural targeting domains so that we can change its tropism. In this work, we used two variants of this exotoxin, the PE40 and even smaller variant, low PE, that has reduced immunogenicity due to lack of the majority of the domain 2 and the point mutations in the domain 3 that reduce its immunogenicity. The mechanism of action of such proteins can be illustrated with DARPin PE40. It binds to HER2 on the surface of the cell, then it is efficiently internalized and goes to endosome, in which the furin protease would cleave the protein and release the toxic module. Then, due to retrograde transport signal, it would go to endoplasmic reticulum, from which it escapes to cytosol. The exotoxin catalytic domain is capable of ribosylation of a eukaryotic elongation factor 2, so it leads to translation inhibition and finally to cell death. So we constructed two proteins, both containing of targeting module DARPin 929 and effector module either PE40 or low PE. Both of them demonstrated very favorable features in vitro because the toxicity established as IC40 was in the picomolar range, 0.1 for DARPin PE40 and 16 picomoles for DARPin low PE. The predominant mechanism of cell death, of cancer cell death, was apoptosis. We can prove it with cell membrane integrity and early exposition of phosphatidylserine on a cell surface. Also, we could observe the DNA fragmentation and the cell morphology typical for apoptotic cell death. The apoptosis is thought to be the most favorable type of cell death for cancer therapy because it does not cause inflammation in the tumor. So, as both proteins demonstrated rather favorable features in vitro, we started to investigate their work in vivo. Unfortunately, the derivatives of Pseudomonas exotoxin A have some unwanted features, like high immunogenicity and considerable site toxicity. So we tried to compare both proteins in this situation. We made two repetitive courses of protein injections on healthy immunocompetent biopsy mice. During all the treatment, we tracked the signs of cytotoxicity, namely the cardio and liver toxicity, with the help of aminotransferase activity in serum. We also established cytotoxicity with the leukocyte count, and the immunogenicity was established as antibodies titer. And after the end of the experiment, we also made a histology study. So we found that DARPin PE40 did cause cytotoxicity. We can see it by the degrees of body weight mice. On the histology level, we could observe the edema in lungs and necrosis points in kidneys. As for DARPin low PE, in the same dosage, it did not cause any significant changes in tissues and organ histology, but when we use twice higher concentration, 20 microgram per injection, we could see some signs of the same toxicity. Nevertheless, they were lower than in case of DARPin PE40. We could not detect any significant heart damage or liver damage, either by transferase activity or by histology study. And as for immunogenicity, indeed, we got what we expected. The DARPin low PE demonstrated lower immunogenicity, and DARPin PE40 did cause the production of high titer of antibodies, and we could even see the proliferation response in spleen. Nevertheless, we tried to investigate the features of both proteins in vitro and xenograft tumor models, and we can see that both of them significantly decrease the rate of tumor growth. DARPin PE40 was tested on subcutaneous inoculated adenocarcinoma cells, and it did cause the decrease of tumor growth and even inhibited it so that the tumor did not started to grow after the end of the course. DARPin low PE was tested on another tumor model, the module of ovary cancer metastasis, and in this case we used fluorescently labeled cells that were injected intraperitoneally, and the rate of tumor growth was established as fluorescent signal in vivo imaging assay. So we can also see that DARPin low PE decreased the speed of tumor growth. At this point of the work, we can conclude that DARPin 929 retains its affinity and specificity to HER2 receptor. It can be used as a component of fused protein. It is efficiently internalized, and this feature can be used for delivery of exotoxin into the cytoplasm. And as for immunogenicity, the combination of DARPin 929 with low PE is better than DARPin PE40 with the same DARPin. Thank you very much for your attention, and I would be happy to hear your questions. I just want to thank all my colleagues from different institutions, mainly, of course, the Institute of Bioorganic Chemistry, and also the Prokhorov General Physics Institute and Lobachevsky State University, with where the xenograft assays were made. Thank you very much.
Thank you very much, Olga, for this very interesting talk. And I would just like to say that it was interesting to see the correlation with internalization assays that you did, the results that you did, and the results that we obtained seem to correlate. I think it was quite interesting.
Yeah, I was also happy to see the correlation between our results. Of course, I've read of it, but the pictures with comments was very effective, very impressive.
Would any of the participants have questions?
Does TNT have any question?
Actually, I would like to read more about these articles and try to learn something from all those published papers.
Olga, we are pursuing now two tracks, both derivatives of Pseudomonas toxin and DM1, the antitubulin drug. And you are now probably the most experienced person here in the DARPin-mediated drug and toxin targeting. What is your spontaneous reaction? Which one variant would be more efficient?
Well, speaking about efficiency, the PE40 is thought to be more effective because even in our experiment we have seen that its IC40 is lower. So maybe it would be more effective. But as for in vivo assays, the low PE is better because, well, as I have already mentioned, it has lower immunogenicity, so it is less restricted by antibodies, by neutralizing antibodies, and it has lower cytotoxicity. It was also shown by other groups that study these proteins. But it largely depends on the way of delivery. We have some works in our lab when we use PE40 in the liposome form. So it is protected from any antibodies. It is hidden there and it can be delivered with no interference with immune system. In this case, maybe the PE40 would be more effective.
If you compare with maytansine derivatives, what would be or your consideration?
Well, I'm not sure, but I'm not sure, but if I remember correctly, the maytansine internalization and escaping the cytosol is less efficient than in case of the derivatives of exotoxin, Pseudomonas exotoxin. So maytansine is also very efficient, but if we speak about low concentrations, and in vivo we would speak about it, the Pseudomonas exotoxin derivatives should be more efficient.
Okay.
Well,
right.
Of course, we should compare in one experiment.
We should compare that in vivo head-to-head, absolutely agree. All right.
I have a question. You showed in your slides that DARPin low PE at 20 microgram doses produced some damage to normal lung tissue, normal kidney tissue. Do you have any comment on that? Do you think this damage is repaired or could be repaired, or how critical that damage is?
Well, if we judge of this damage as in terms of general condition of mice, we could not see any difference. They did not lose weight, they did not lose some, I don't know, appearance, the fur, everything was okay. So maybe it was not very critical for them. We know by the clinical trials that involved Pseudomonas exotoxin derivatives that these side effects are quite common, and in case of patients with severe disease, they can be limiting. At least we know about these side effects in case of PE40 and PE38, very similar derivative. And there was one trial, one clinical trial in humans that used low PE, and in this case, the same side effects were very moderate. Sometimes they could not be even detected in the dosages that were used for therapy. So I can say that these side effects do exist, unfortunately, but in the dosages that are used in therapy, they are tolerable.
Thank you. And probably there is also a balance between the anti-tumor efficiency versus the anti-tumor killing efficiency versus the side effect, where for targeted therapeutics, it's still much lower side effects compared to non-targeted chemotherapeutics.
Yeah, of course. So it is known that the exotoxin, the third domain of the exotoxin, has the same cytotoxicity with no regards to the targeting module. So these unfavorable features are the features of the exotoxin. And introduction of the targeting module, of course, reduces this, but does not bring it to zero. And the low PE variant was shown to decrease this effect, this interaction of untargeted protein with lungs and kidneys. So, of course, the targeting module decreases the side effect, but we should also consider the effector module because it has some side activities. Fortunately, we can map the sites of this interaction, and we can decrease it somehow. That was made for low PE.
Do I ask one question?
Yes, of course.
Where did you do your blood analysis?
Well, we did it in our lab. We used the flow cytometer for this assay. So actually we did 2 assays. We made the Giemsa stain of the blood smears, and also we did flow cytometry analysis on Novocyte cytometer. It is used in clinical practice, so we can calibrate it to analyze mice blood. So we did.
If we will be able to send you once upon a time blood, because our veterinarian university in Sweden cannot do this.
Alkaline phosphatase is of interest.
Yes.
First of all, creatinine, alkaline phosphatase, bilirubin. So these
I see.
blood markers of toxicity.
Oh, now I see. As for ALT/AST analysis, we did it ourselves. We just bought the, the kits that can be used in clinical practice, and just calibrated them for small amounts and did the whole assay. So this analysis can be done in our lab. As for other biochemical points, we did not tried it, so I'm not sure we're quite experienced in it, but we can try.
Okay. Apparently I experienced enough. Okay, very good.
Would anyone have any further questions to Olga?
Could I ask a question?
Yes.
Yes. So there is a slide that you show the comparison of the animal study, but one of your experiments, you said it was IP injection. Was it the?
It was, yes. Probably I should not stop the screen. Well, okay, I would say it in words. In our one of our studies used the ovarian cancer metastasis model. So we injected the cells in intraperitoneally, where usually the ovarian cancer cells make metastasis. And the tumor, well, actually the disseminated tumor of a lot of metastasis formed. And then we injected the protein intraperitoneally, because in clinical guidance, we've read that in cases where when it is applicable, the intraperitoneal injection of the therapeutics is better. It is preferable because it decreases, the site toxicity, decreases the sometimes even immunogenicity. Well, it helps to provide higher concentration of the therapeutics in a site of the cancer growth. So we just tried to make the most relevant to clinical situation test.
Yeah, it was very interesting because we used the local tumor model and we inject by IV. Then we observe some, maybe the toxin infection to the vessels of the tail. So this is quite interesting.
The vessels effects are a big problem and it is quite common. Yeah.
Good way to label the substance and compare to different way of injection head to head in mice. We can do it, right?
Quite interesting.
Small cute study.
Yeah, we should think about that. The biodistribution is an urgent feature of the therapeutics.
Would anyone have any concluding remarks or any further questions? I think it was a really great forum and very, very interesting presentations. And I hope we can continue in the future. Definitely some more clinical results from Tomsk that could come soon from DARPin, and of course from Uppsala, and hopefully from Moscow again.
Well, I'm very happy that Olga was here with us, and I could find definitely further direction of our collaboration and possibly help us to set certain assays, which are definitely set up in Moscow. So we should probably communicate on this topic, definitely. We have this forum recorded. I hope that, Maria, could you please arrange a Dropbox for forum participants? And I would like also all speakers to ask to send their presentation to this Dropbox, so we could once again to go through the slides and through the presentations. It's a great opportunity nowadays. All right.
Thank you very much to all participants and everyone who joined us today.
I hope that I like very much what I have seen today, and it's some discussions, and that was the aim, to have direct contacts first. And second, we have a certain idea to keep such forum probably 3 times a year. So next time, hopefully sometime during late February, and we have more exciting results to share, and let's keep in touch. I think together we're doing interesting work.
Okay, maybe I would like to add a couple of words. Today it was organized a very good agenda. We have two talks about radioactivity materials and about toxins. And I am really not modest, but happy to say that today our paper is accepted in ACS Nana journal. And in this paper, we demonstrate that dual targeting strategy is drastically enhanced anti-cancer therapy, which made possible 100,000-fold decrease of effective drug concentration. And in this paper, we used low P toxin, which about what Olga Shilova told now, and antibiotics. But we can use the same approach using the radioactive materials and this toxin based on drug DARPin targeted drug delivery. What actually was interesting that this dual combination DARPin-based toxin made overcoming tumor escape. I guess it would be a very interesting point of our further efforts to combine these radioactive materials and toxins, a different origin like pseudomonas toxin or metenzine. And I would like to stress that this combination, which was demonstrated our today agenda, radioactive materials and toxins, could be really. It's my vision of today talks. Thanks.
Thank you, Sergey Mikhailovich. Once again, I just would stress that we share the vision. Right now, we're running two parallel studies, and one is co-treatment of SKV3 tumors with trastuzumab and radionuclide pre-targeting antibody and antibody. And second, we're struggling with co-targeting of trastuzumab and EC1 low P. So different target and the same, but we're setting up this technology. It's also logistical of experiment and so on, which is not always that easy. But I'm sure that we will go your way, and we will combine probably toxin and radioactivity in the near future. So that is on agenda. Not sure that we will be completely ready by next forum to present the results, but the forum after next, for sure, we will have. And also clinical results are piling up, and I'm extremely happy to see that there is a great progress with DARPin G3 study run by Olga and Vladimir, and they company. I think Tom's could show also very exciting results with deoxyglucose, technetium labeled, and so on. So we have full agenda for next meeting.
We are definitely on the right way.
Just want to express that I'm personally very, very happy to work with such a team. Thank you very much.
Thank you very much to all participants and everyone who's been here, and also to the presenters. And I hope that we will meet probably next year, early next year, once again.
All right. Thank you.
Thanks a lot for everybody.
Thank you, everyone.
See you next time.
Thank you.